Looking at the small intestine, we can first divide it into its three regions. The duodenum is about the first 1 foot. Duodenum means 12 fingers, so they first measured the duodenum using fingers. As you can see, the duodenum's primary function is to receive the chyme from the stomach, but also neutralize the acidity of that chyme. And we already learned that that neutralizer is bicarbonate that comes from the pancreas. So the duodenum receives chyme, neutralizes the acid. Then the next 3 feet is known as the jejunum. And jejunum means empty, because when they first opened up the jejunum, it was empty. It didn't have material in it. The last 6 feet, so the longest region of the small intestine, is known as the ileum. So we have the 3 regions of the small intestine. The first and most important thing to remember about the small intestine is that due to its large surface area, it does most of the absorption, but also most of the digestion along the digestive tract. You'll notice inside the small intestine there are many circular folds. These circular folds are one reason why the small intestine has such a large surface area. These circular folds also help almost spiral the material through the small intestine, and this allows a longer contact time between the wall of the small intestine and the material. This allows proper absorption of the nutrients. Looking at its histology, you'll notice there are other structures that increase the surface area. There are these finger-like structures known as villi, and then along the edge of a villus, there are very, very small microscopic structures called microvilli. So together you have the circular folds, the villi, and the microvilli, all contributing to the large surface area of the small intestine. You'll notice located inside each villus, you're going to find blood capillaries that are going to be receiving nutrients after absorption. You're also going to find lacteals. I'll remind you, lacteal means milky. A lacteal is a lymphatic capillary that absorbs dietary fats. And when those dietary fats enter the lymph, it makes the lymph look a little white or milky in appearance. The submucosa isn't all that different. It's got connective tissue. You'll notice some blood vessels. It's got some neurons or nerves. The muscularis goes back to 2 layers of smooth muscle, a circular layer as well as a longitudinal layer. The neurons controlling those muscles are the myenteric plexus. And then the outer layer is that serosa. So the major changes here are the microvilli, the circular folds, and the villi, as well as going back to 2 layers of smooth muscle. This is a real micrograph showing the villi, as well as the fact that there's a number of goblet cells. So these almost clear looking cells lying along the epithelium in between the absorptive cells. Absorptive cells are the names of the cells that do most of the absorption along the small intestine. These goblet cells are secreting mucus. And we often also refer to the lining of the small intestine as the brush border. The brush border because the small little microvilli under high magnification look similar to a hairbrush. Looking at the absorption, so this occurs after we've done all of our chemical digestion. You'll remember that a lot of the enzymes that have been doing this chemical digestion in the small intestine came from the pancreas. There are a few enzymes produced by the small intestine. They include lactase, which is going to digest lactose, sucrase, which is going to digest sucrose, and maltase, which is going to digest maltose. Those enzymes complete the carbohydrate digestion. So once carbohydrates are completely digested, they're down to the monosaccharides, which include glucose, galactose, and fructose. Those monosaccharides eventually get absorbed in through the absorptive cell into the blood vessels, into the blood capillaries of the villi. Then eventually they go to the liver via the hepatic portal vein. Proteins, when they're done being digested, they end up being mostly amino acids. Maybe you get a couple dipeptides and tripeptides, which are just 2 amino acids or 3 amino acids. They get absorbed into absorptive cells, and through the absorptive cell into the blood vessels, and eventually the hepatic portal vein and the liver. Short chain fatty acids, which are just very, very small fatty acids that have been digested off of triglycerides, they go into the blood vessels as well, the blood capillaries, and go to the liver. But you'll notice the big difference here is with the long chain fatty acids. They actually get reassembled inside of an absorptive cell. So notice we have the fatty acids and the monoglycerides, which is basically a single fatty acid attached to a glycerol backbone. They're going to be absorbed in. Triglycerides are reformed inside the absorptive cell and packaged in a lipoprotein called the chylomicrons. A chylomicron contains triglycerides, so dietary triglycerides, the triglycerides from your diet, but also some proteins to help those triglycerides dissolve better inside the lymphatic capillaries. So it's those chylomicrons that enter the lacteals and then eventually drain into our bloodstream via the thoracic duct, which is attached to the left subclavian veins. So there's a big difference here between absorption of monosaccharides, amino acids, versus fatty acids. This is also shown in this picture, where you have a drawing of a villus inside the lining of the small intestine. We're getting absorption of amino acids, monosaccharides, and short or small short chain fatty acids, just the teeny tiny fatty acids. They go into the blood, eventually go to the liver via the hepatic portal vein. They get processed, either stored or then released into the bloodstream via the hepatic vein. Here we show the chylomicrons containing the larger fatty acids repackaged with proteins so they can be transported in the lymph. That lymph will eventually go to the thoracic duct, and that thoracic duct will eventually deliver those triglycerides into your circulation at the left subclavian vein. Finally, the small intestine has to absorb other things. So at this point, we've talked about absorption of monosaccharides, amino acids, and fatty acids, or triglycerides. The small intestine also does quite a bit of water absorption, as well as minerals and vitamins. So if we look at water, or fluid balance, On average, you ingest about 2.3 liters. So that's the water that you drink, water that's in your food, that enters the GI tract through ingestion. You secrete about 7 liters into the GI tract. So this water comes from saliva, about 1 liter a day, gastric juice, about 2 liters a day, 1 liter of bile, 2 liters of pancreatic juice, and 1 liter of intestinal juice. So this is all the water other than the water you ingest that goes into your GI tract. Now that's a lot of water. And we don't want all that water to leave the GI tract. So we need to absorb a lot of this water. Most of this occurs at your small intestine. 8.3 liters gets absorbed at your small intestine because of the high surface area. The large intestine is going to also absorb some water, about 0.9 liters, so just under 1 liter of water gets absorbed at the large intestines. And that leaves about 100 milliliters or 0.1 liters that actually gets excreted from the GI tract. And that's an important water to have in feces because you definitely do not want feces to become dehydrated because that can contribute to constipation. And a number of treatments for constipation are drugs that leave water in the GI tract. So it blocks a little bit of this water reabsorption. Therefore, you get a little more water in the feces, which helps with constipation. This is a good summary table of digestive activities, which include involvement of the pancreas and its enzymes and buffer, involvement of the gallbladder, primarily responsible for bile storage and delivery, but also the small intestine and the number of functions dedicated to the small intestine. In the next video, we will start our discussion on the large intestine.